Finally, I want to talk about the cerebellum. Cerebellum is this almost small little structure at the back of the brain, which looks very distinct. The brain and the neocortex has gyri and sulci, whereas this looks very different when you look at the specimen. And you can go to King's College. They've got a laboratory, a museum rather, of these sections, prosections, and you can see how the cerebellum looks very different. There's no sulci and gyri, but it's got these horizontal lines, and it's connected. It's very close proximity to the brainstem, in particular the pons. There are three connections, which they're structural and physical connections called peduncles. Superior meaning the top, middle meaning in the middle, inferior meaning at the bottom. So there's a superior, inferior and middle peduncle which fix the cerebellum onto the brainstem. And what they do, the cerebellum is mainly involved in movement and coordination and posture. So the fact that you're able to stand up straight and walk around is down to the cerebellum. Okay? So if someone's got ataxia, which means that they're walking around funny, yeah, you either think they're drunk or you think about a cerebellar lesion. Okay? So that cerebellum is very, very important for movement. Medical students and doctors have a mnemonic, Danish, which stands for some very funny words like dystidocholkinesia, ataxia, nystagmus, intention tremor, slurred speech, and hypertonia. If patients exhibit signs of these signs, then we're thinking about a problem in the cerebellum. Dystidocholkinesia sounds remarkably complicated, but it's very easy to pick up. All we do is we ask a patient to, and hands up if a doctor's ever asked you to do this, just to tap your hands like this. Have you ever had that done before? Can you all do this for me? Is anyone not able to do that? I do know a good hospital around here if someone's not able to do that. Okay, and now on the other side, just check that both of the cerebellum are working. Good. That ability to do a repetitive to and fro motion is the cerebellum working. If you're not able to do that, that is called dystidocholkinesia. Okay. So next time you hear the doctor say that in A&E, there's no dystidocholkinesia, you can say, yeah, I know my cerebellum's working fine. And you can really make him work extra hard and thinking that he's maybe got a professor of neurology in front of him. Ataxia means that you're not able to walk in a straight line. Nystagmus is the jerking of the eye movements. Intention tremor means that when you do something, you then develop a tremor. So normally you're okay. But as you go to do something, whether it's to pick up a pen, you then develop a tremor. And that's an intention tremor. Compare that to a resting tremor, which is what Parkinson's disease patients have. So they're normally resting, and they normally have a unilateral tremor like this. It's called a pill rolling tremor because their finger is moving, their thumb and their finger are moving in this way. You might have relatives who've got Parkinson's disease, and that's a resting tremor. And sometimes that's obliterated when they do something, whereas an intention tremor is the opposite, where when you go to do something, the tremor comes on. Slurred speech is a sign of disease here, and hypotonia, that's when you become floppy and flaccid. That's also a sign of cerebellar disease. Just a couple of other brain structures that I want to mention very quickly. Amygdala and cingulate gyrus. So these are underneath the cerebral cortex. Amygdala is involved in memory, decision-making, emotions, aggression, and fear. The amygdala is very important in fear and aggression. Some studies have found that people who are very aggressive have larger amygdalas than others. And sometimes in rush hour, it seems like most people have just got an amygdala rather than a brain. But that's a separate study in itself. But that is the key structure involved in fear. There are some people who have a problem in the amygdala, and so they're not able to sense fear. And that's obviously dangerous. If you're not able to get away from a dangerous situation, that can cause harm to life. The thalamus is this structure here, which just looks like half an egg. Looks like half an egg. And that's involved in integrating all of the information, receiving all of the sensory information coming to the brain from the periphery and the spinal cord, and also integrating all of the cerebral information coming from the cerebral cortex going down to the muscles. So it integrates all of the information that the brain is receiving. Now, I purposefully said that it looks like half an egg. And it really does. But the thalamus itself is one of the most remarkable neuronal structures that we have. And if you're interested, there's a book for £399 just on the thalamus. And somebody guess how many pages that book has? 500. That's a very big book. We're talking just about the thalamus. It's got more than 1,700 pages just on the thalamus.
Is that the cost of it, or how much it weighed?
Yeah. Well, the cost is £399. Well, actually, there's a £41 off, actually, if you buy it this month or next month. So, light at the end of the tunnel for the thalamic experts. So that's the reason I mention that is the neuroscience is just fascinating. And the deeper you go in its ocean, the more pearls you'll get. But nobody really understands it. We're only just putting it together now. Hypothalamus and pituitary, I'm going to talk about this in great depth in the endocrine lecture, because this is really the control of human physiology, hormones and things like that are all regulated by the hypothalamus and the pituitary gland. The hypothalamus sends hormonal stimuli to the pituitary. Pituitary then releases hormones in a circadian rhythm. That means that it's a very set rhythm to do with your day and night pattern. It's important right down to when you became adults. It was the pituitary and the hypothalamus which made you enter puberty by sending out the right signals to release the right amounts of hormones. And the pituitary, if that thalamus looks like an egg structure, the pituitary is just this tiny structure here. I don't know if it's projecting well at all. But if there's a problem in the pituitary, that can actually be fatal. So if the pituitary infarcts, i.e. does not receive oxygen, then a patient can die very, very quickly. And there are conditions which cause pituitary hemorrhage. So in fact, even obstetricians look out for this condition. If a woman gives birth and then she has a major hemorrhage afterwards, the pituitary vessels are very sensitive and they can constrict as part of a blood-saving mechanism. And that constriction of blood around the pituitary can decrease the supply to the pituitary, and therefore you don't get a release of key hormones such as ACTH, stop cortisol, and that can crash your blood pressure down into dangerously low and put you into a hypovolemic cardiac arrest. Very important structure, although very tiny. Just looks like a pea, a garden pea. And finally, the hippocampus. The hippocampus is so named because it comes from the Greek hippos, meaning horse, and campus, meaning sea monster. It looks just like a seahorse. And it's a bilateral structure, and it's involved in memory consolidation and formation. So all these good memories that you have of your childhood, and I hope you have many, were consolidated by the hippocampus. There is another important case that I want you to be aware of when it comes to the hippocampus. Does anyone know the story about Henry Molaison? Henry Molaison was always known as HM until 2008, and that's because he died in 2008. He's one of the most widely studied human beings in terms of hippocampal pathology. This is a man who, when he was a boy, about 8 or 9 years old, he fell off his bike, and since then he developed epilepsy. By the time he got to 16, his epilepsy was very, very severe. Are we all familiar with epilepsy? It's the seizure condition which makes you, gives you seizures at any time. It got so much that he went through all the neurologists of the time who put him on every single anti-epileptic medicine known to man, titrated it to their maximum dose, but still he was having lots and lots of seizures. And he was very, very young. So in the end, he got referred to a neurosurgeon. And that neurosurgeon was called William Beecher Scoville. Scoville did a series of tests on this man and found that the majority of his epileptic foci are from his temporal lobes, in particular the medial parts of the temporal lobes. And he offered him a procedure. He said to him, I'll remove both parts of your temporal lobes, in particular the medial components of them. And what the surgeon did, he did just that. He removed the hippocampi, the hippocampus on both sides, the parahippocampal cortices, the entorhinal cortices, the piriform cortices, and the amygdalae. These are all very close to the medial temporal lobe. And what they found is, over time, as they started to scan him, is the remaining little bit of hippocampus that he had, or hippocampi that he had, also atrophied because they were getting no stimulation, so they just essentially disappeared. So he was a man now without a hippocampus. And what they found, he was completely unable to make new memories. And this formed part of our understanding about the importance of the hippocampus in memory formation. They found that he was able to remember his previous memories, so it's not necessarily the store of memory. The store of memories we know mainly around the cingulate cortex, but this is about consolidating memory. So it's a very important structure to be aware of. And this is a very interesting case because he's very topical in medical school application interviews. It's something that you can mention. So they are the brain structures. I know that was quite taxing on the brain, and I hope you will use your hippocampus to consolidate some of that information later when you go home. But it's a very fascinating structure.